In this topic, we'll be discussing protein primary and secondary structure. So I will be going over some of the key features of these levels of protein structure and what key components you need to know regarding each one of these structural aspects of proteins. The learning outcomes are as described below. So we consider proteins in a cell. They have many diverse functions. You don't need to memorize this list, but we'll be going over these as we proceed through the course. They function as enzymes to catalyze biochemical reactions. So reactions that would have a half-life of 1,000,000 years, you can have 1,000 events occur in 1 second with an enzyme catalyzed reaction, just to show you the ability to enhance rates through enzymes. So we'll be talking a little bit about that in another topic. You can use proteins to store and transport other molecules, so they can act as reservoirs to store ions and then release them in a controlled manner, as well as transport molecules across membranes. They can serve as channels across membranes as well for different types of specific ions and cofactors. They can also serve as structural components of the cell and organelles and tissues. So our muscles and our nails and our hair are composed of protein, as an example. So we're talking about some of these structural proteins in a different topic. They can also participate as mechanical motors to move cells and transport things across membranes as well. We'll be talking about that in terms of ATP synthesis in a later topic. They can be receptors for extracellular signals and transduce those signals into the cell, and that will also be another topic. What we won't cover, but is important, and we'll have some examples here, but the courses for principles of biochemistry as opposed to trying to do everything in your textbook. So in terms of gene expression, transcription, and translation, that will be touched upon in the context of what we cover in the course, but not as specific topics. And so those are reserved for other courses. If you take molecular biology, for example, which you should have already in some of your previous courses, you will have covered those topics there. We offer it as part of our biochemistry major, a specific course related to gene regulation of gene expression, but that's a different course, and we would just do not have time to go into that, but they're involved in those processes as well. So proteins, because they're involved in diverse functions, they themselves are very diverse, but there are structural aspects of proteins that are shared with proteins in general, and we'll be talking about those structural components in the next several topics. The levels of protein structure can be defined as primary structure, which is shown here, just the sequence of amino acid residues. The secondary structure, which are local folding events that are held together by hydrogen bonds. And so we will be covering these two particular topics here in this topic presentation. The tertiary and quaternary structures are higher order structures, and those will be covered in a different topic. So you'll see this slide again in the other topics related to tertiary and quaternary structure. Right now we're going to focus on the primary and secondary structure in this topic. Just wanted to remind everyone of the non-covalent associations that are possible. We can look at a charge-charge interaction between two charged species like a protonated amine and a deprotonated carboxylate. So you can envision some amino acid side chains that can participate in this type of interaction. A charged dipole, where you have a charged molecule and a molecule that has an overall dipole, like for example water is shown here. The dipole goes across the molecule this way. This is the positive end of the dipole, and the arrowhead is the negative end of the dipole. Those dipoles will align to best interact with this charge. There's dipole-dipole alignments, where dipoles will align according to their partial positive and negative charges. You can have a charge induce a dipole on a molecule, like an example would be the side chain of phenylalanine and the side chain of lysine. This would be a charge-induced dipole interaction. A dipole can also induce a dipole, like water, for example, on a phenylalanine ring, but this is going to be very weak as you go down the list from the top to the bottom. The interactions get weaker. Dispersion forces, like the van der Waals interactions, for example, where you have mutual synchronization of fluctuating charges, where you have the electrons in two molecules are synchronized in such a way to provide a partial ability to interact, but very weakly. But if you add a lot of these forces together through many features of a protein as it folds, it does become a significant interaction because you have so many of them. So it does contribute to protein stability, and we'll be talking about that in this topic and in other topics in the protein sections. And the last is hydrogen bond, where you have a donor and acceptor. The donor donates the hydrogen, the acceptor accepts the hydrogen through lone pair interactions with that hydrogen, forming a hydrogen bond. And there are many functional groups present on biological molecules that can do hydrogen bonding. So this is a very prevalent one and very important when we're talking about secondary structure formation. So this is review, so you should have a firm grasp of these interactions because they'll come into play in forming these higher order protein structures. So as mentioned previously, the primary structure of the proteins is the amino acid sequence. So the amino acid sequence is written down from the N terminus to C terminus, and there are experimental ways in which we can determine amino acid sequence from proteins. We just do not have the time to go into those experimental details in this course. So just remember the primary structure is the protein amino acid sequence. So if you're asked, what is the primary structure of a protein? The answer is it's amino acid residue sequence. The secondary structures are what we're going to focus on in the remainder of this topic. And we have to consider the chemical features of a peptide. In this case, you want to consider a polypeptide that has many amino acids in it. And you have to consider the aspects related to the actual peptide bond itself, because that is what's going to be driving the ability to form these secondary structures. So if we look at a segment of a peptide where we have amino acid residue 1, amino acid residue 2, and amino acid residue 3, you can see the different side chains here. Here's your alpha sub 1 carbon, and your side chain R1, alpha sub 2, R2, alpha sub 3, R3. These are all L amino acids, and they're hooked together. The alpha carbons the carbonyl carbon, carbonyl oxygen, amide nitrogen, and hydrogen are all in a plane. So recall that with a peptide bond or an amide bond, there's a lone pair of electrons on the nitrogen in a p orbital that can resonate with the pi bond of the carbonyl group, and you have delocalization of this lone pair of electrons across these atoms, thus making the carbon-nitrogen bond of the amide bond, the peptide bond, have partial double bond character, and that we favor the trans conformation, and that's defined as the alpha carbons being trans along this amide peptide bond, and that these all these atoms are in the same plane because everything is sp2 hybridized, except this carbon is sp3 hybridized, but the carbon itself is in the same plane as the sp2 carbonyl carbon and sp2 amide nitrogen. And so that's shown here as a sheet. And so you can think of this as a piece of paper, and that there's freedom of rotation along this carbon-carbon bond between the carbonyl carbon and alpha carbon, and the same with the amide nitrogen and alpha carbon here. There's rotation of these planar regions along the backbone. So there's not going to be any rotation in the amide bond or the peptide bond itself. No rotation here, but there is rotation here and is rotation here. So these things can start to fold up and rotate. And when they do that, they present some unique opportunities for hydrogen bonding. Again, remember that the alphas are always going to be trans along the peptide bond. So you can see here this peptide bond, alphas are trans. This peptide bond, alphas are trans, and so on. So based on these chemical features, these polypeptides will start to fold into more higher ordered structures. And we're going to talk about the secondary structures in this topic. So to understand better these structures, there's ways to define the atoms in three-dimensional space. So every atom will have a x, y, and z coordinate. But there's also the ability to look at the angles in which these planar structures can rotate along these alpha carbons. And so by definition, the angles are defined as shown. So between the nitrogen and the carbon, that's your phi angle. Between the alpha carbon and the carbonyl carbon is the psi angle. And between the carbonyl carbon and the amide nitrogen of the next amino acids part of that peptide bond is called omega. But as we stated before, there is no rotation around omega because of the partial double bond character. You have to pretty much have an enzyme to help facilitate that rotation. And so the alpha carbons will be trans along the omega. And so these planar regions then by definition are going to be in the same plane, if you will, because of the rotation of this bond, as we'll define these dihedral or torsional angles. And so as a result of this, this hydrogen and this carbonyl oxygen are in the same plane, but they're 180 degrees rotated from one another, and so that hence the omega angle is 180, and that's it. It's always going to be 180 because of this trans configuration here of these alpha carbons. So that angle is going to be 180 degrees. In the case that's shown here, psi and phi angles, this would be your psi angle, and this is your phi angle. They will vary based on the secondary structure, and there's going to be certain ranges of angles that are possible based on the secondary structure that's formed. So some angles will not be permitted, and you can see that here in this illustration, where if you rotate too much around this alpha carbon, you start having steric clashes with the substituents that are attached to the carbonyl carbon. So the amide hydrogen here is starting to clash with the carbonyl carbon oxygen of a neighboring residue if you over-rotate. So not all angles are possible under these psi angles because of these steric clashes that occur. And if you think about the side chains, they would also have a possibility to have steric clashes under certain angles of rotation. So you have to kind of keep in mind the angles here will not be a 360 degree available space, but restricted. So these are the torsional angles, but omega is always 180. So you may recall how to define what a torsional angle is, and so you have to look down the axis of the bond upon which you're looking at rotation, and you can define the rotation based on plane. So for example, if we look at this substituent here and its position down this bond, this sigma bond we're looking at, this covalent bond, to this other substituent on this atom here. So think of this as a alpha carbon, and say that's your carbonyl carbon there. You can look at a substituent on the alpha carbon relative to the carbonyl carbon, and if it were in the same plane, if it were part of this orange area and was vertical here, that would be your angle, would be 0. If you were to rotate it, in this case it's showing omega, but you could imagine it could be phi or psi as well. How much it rotates will be positive, or it could rotate the other direction and be negative. So if omega is 180, plus 180 and minus 180 is the same position, it would rotate all the way down here and be anti relative to this bond. And so that would be 180 degrees. And so values can change, either rotating to the right like this or to the left, it would have a negative value. So you have to define this angle, and it takes 4 atoms to do so. 1, 2, 3, 4. Okay? Clockwise, positive value, and counterclockwise, negative value. This is the torsional angle. So know what a torsional angle is. So let's look at our first secondary structure, and it's called the alpha helix. So the structure is shown here in ball and stick diagrams. The planes of the peptide bonds are shown here, and you can see how they're kind of arranged in a helix fashion. This isn't necessarily the best picture to see that, but what I do like about this diagram is that you can see the hydrogen bonding interactions. So what you have are hydrogen bonding interactions between an amide hydrogen that's attached to this nitrogen, hydrogen bonding to a carbonyl oxygen of a residue that's further down in the sequence. And notice here's another one up here that's hydrogen bonding. Here's another one over here. Here's another one here and another one here. So you have all these amide hydrogen, carbonyl oxygen, hydrogen bonding situations occurring with residues on this strand in such a way it gives a helical structure. All right. In fact, when we look at these structures, we find if this were amino acid number 1, it hydrogen bonds to amino acid as the 4th one down. So we have an I to I plus 4 hydrogen bonding. So if this is number 1, this would be number 5. And so it hydrogen bonds as shown. The amide hydrogen bonds to the carbonyl oxygen. And then this amide hydrogen here, hydrogen bonds to the I plus 1 down here. So every carbonyl oxygen and amide hydrogen is hydrogen bonding between the I and I plus 4 residue to form this alpha helix. So all the hydrogen bonding occurs. There is no amide hydrogen that's not hydrogen bonding in an alpha helix or carbonyl oxygen. So this has nothing to do with the side chains. The side chains actually stick out. These stick out into solution. These R groups face outward. And I got some other pictures that will show that better. If you do some measurements here, we find that the difference between the turn of a helix, a complete turn where you can kind of rotate 360 degrees and get back on the same position on the face of the helix, is 3.6 residues per turn. So it's not exactly 4 like the hydrogen bonding pattern, but it's 3.6. And the distance between those is 5.4 angstroms. If we go through here and measure these phi-psi angles, these dihedral angles or torsional angles, you'll find that for an ideal helix, the phi angle is minus 57 degrees and the psi angle is minus 47 degrees. And shown here is the C-terminal end versus to the N-terminal end. All right? So this is the detailed structure of an alpha helix. I don't want you to memorize these numbers, but I want you to key on the key features is that the hydrogen bonding is between the I and I+4 residue. And if you remember 3.6 residues per turn, you're good to go from this diagram. We're going to look a little more detail here on some other aspects of the helix in the other slides. But from this one, remember it's the I to I+4 hydrogen bonding, 3.6 residues per turn. Another feature of the helix is that the helix itself also has a dipole moment. So just as we've seen with smaller compounds that have heteroatoms attached to carbon atoms, where you have dipole moments along these connected atoms that can give rise to an overall dipole moment on the molecule. We also have the dipoles of the peptide bonds that are present in a polypeptide chain that, when folded into a helical structure, it confers a dipole moment on the helix. And so recall, here is your peptide bond, and it's in the trans configuration, so your omega is 180 degrees here. Lone pairs in resonance. And so when we look at this peptide bond, shown here's your nitrogen, carbonyl, oxygen, and carbon, there's an overall dipole moment here across this peptide bond, as shown. So you have a delta plus end and a delta minus end. When we overlay these dipole moments across the peptide bonds in a helix, they all point in the same direction, from the N-terminal end to the C-terminal end. You can see these little dipoles from the peptide bonds point in the same direction. And the result of that, there's an overall dipole moment on the helix that goes along that helix from the amino terminus to the carboxy terminus. When we look at the implications of what this means, well, you can imagine that if you had a phosphate and it was going to bind to one end of a helix here, it would be more attractive to the positive end, I should say, because it would be negatively charged, and we'd be looking at the interaction with the amino terminus, and that's kind of shown right here. If you had a positive molecule, it would be more attracted to the C-terminal end of the helix. It will favor the dipole. And also we know that the C-terminal end of a peptide or a helix in this case, it may not be terminal in the protein sequence. It could go on to form other secondary structures. And so the dipole alignment of the helices in a structure can kind of help us understand maybe some interactions that may be occurring with other molecules. So it's important to understand this dipole moment. If we were to look at the helical dipole, it could be measured or estimated based on the number of peptides bonds that are present in the helix, and just multiply it by 3-point by units, which is due to the single dipole of a peptide bond. So if we look at a typical helix length and this dBi unit per peptide bond, it's almost as though there's like a half of a formal charge to a 0.7 charge conferred based on this dipole on the end of a helix. So it is substantial and important in how proteins interact with other molecules. Understand the helices do have a dipole moment. When folks start to do more structural studies of proteins, again, Linus Pauling, based on what was known for amino acids and some crystallized peptides at the time, postulated that there should be alpha helical helical structures in proteins based purely on theoretical considerations. This is before anyone has determined any actual structures of proteins. And when they did the first protein structure, they did see helical regions, but they were of the alpha helical type that we described, the interactions between the I and I+4 in terms of our hydrogen bonding. So Pauling did get it correct in terms of this hydrogen bonding interactions with the amide hydrogen and the carbonyl oxygen, but he didn't have the alpha helical pattern as part of his postulated structure, but he did have the pi helix, which is hydrogen bonding interactions between an I and I+5. And those do exist in proteins as well, but not to the prevalence of alpha helical regions. And so you can see here the hydrogen bonding pattern makes the helix wider than the alpha helix. So here we're looking at the space filling model, van der Waals radii of the helix in this direction. This is what's called the ribbon diagram that's shown here to illustrate the secondary structure of a helix in a protein. The directionality here is from N terminus to C terminus. And this is the top of the helix, looking at the top of this helical region. So we're looking at the top of it. This is along the side. And so the alpha helix is tightly wound. There's no hole. The pi helix is a little wider, I to I+5. And because it's wider, it's also shorter and has a little hole down the bottom. In determining more structures, it was found that there's also another helix that hydrogen bonds between the I and I+3, and that's called the 310 helix, and that's shown here. So it's elongated relative to the alpha helix, and you can see here it doesn't have a nice round parameter or structure, but it's more of a triangular shape when you look at the top of the structure. So the perimeter of it is not a circle, but it's kind of like you got these different indentations here based on it being more tightly wound of a structure. So you can see based on these features, you have different structural characteristics of these helical regions. So you don't need to know all three, just know the alpha helix is I to I+4 and hydrogen bonding. All of these would have dipole moments as well. It's just that they would be different strengths based on their hydrogen bonding interactions. But notice all these hydrogen bonding interactions are in the direction of the helical axis. They're not perfectly parallel, but they go in the same direction. And the ones that are a little more parallel will probably be the pi helix relative to the alpha helix or the 310 helix, because they tend to be more angled in that case. So all possible structures of these helical types have been detected in proteins. when researchers started to get more structures of proteins, they started to look at the propensity of amino acids. Are there certain amino acids that would be in helical regions versus other regions of the protein? So the idea being that if a protein sequence is known, can we predict what regions of that protein would be helical or not? In other words, do some predictions based on sequence only, what the fold will be like. So people still work on that today in terms of give me a sequence and I'll show you what your protein looks like. And they're still pretty good at some models of sequence to protein structure, but sometimes the models don't actually reflect the actual structure of the protein. Some other considerations are needed in the protein folding process, and we'll talk more about that in the topic on protein folding. But right now I just want you to think about, well, gee, can any residue be in an alpha helix, or only certain subset of the 20 amino acid residues would be in a helical region? And so once there were a few number of structures and different experiments being conducted in terms of looking at that, is there a way to measure the propensity of an amino acid to be an alpha helical conformation? And so if we look at the change in the change of free energy, kilojoules per mole of a residue that's present relative to alanine. So alanine would be the delta G of itself minus itself, which would be 0, is the basis of everything, based on alanine. Is this residue more prone to being an alpha helix than alanine or not? That's what this delta G is telling you, is illustrating here. So let's look at arginine. It says it's 0.3. So it's a little less able to be an alpha helix than alanine is. So the higher the number here, the less likely it is to be in an alpha helix. If it's negative, it would be better than alanine, but alanine is the best one. Well, leucine's pretty good here. There's a couple that are below 1. They're pretty good, but there's no negative numbers here because alanine's being the lowest. So it's normalized to that one. So the higher the number, the less it is. So a lot of these are close to less than 1. There are a couple larger ones. Look at glycine, very large. What's another large one in here? That's 4.6. Ooh, there's another 4. So you can see there's 2 outstanding residues here that really don't like to be in a helix. Glycine and proline. Knowing the structures of glycine and proline, can you figure out why those amino acids would not be good candidates to be in an alpha helix? I want you to think about that. I want you to think about that and find the answer for yourself, because it's based on those amino acid side chains. So you have to think about the side chains in context of an alpha helix, because remember, the alpha helix has the side chains pointing away from the helical axis, and they're pointing towards the solvent. And the formation of the alpha helix has to do with the backbone, carbonyl oxygen and amide hydrogen bonding interactions. It has nothing to do with the side chain. So how would these side chains kind of cause some disruption in helical structure that would not promote helix formation? That doesn't mean we do not find a glycine in a helix or a proline in a helix, but on average, they're usually not. They're usually not. The other secondary structural component we're going to look at are called beta sheets, and they're very common. And there's 2 types of sheets. There's parallel, anti-parallel. So this is a different hydrogen bonding pattern than we've seen with an alpha helix, the alpha helix hydrogen bonding pattern, is with the backbone residues that are hydrogen bonding between the I+1 residues in a very systematic way. So the residues have to be close enough together in sequence in order for those hydrogen bonding interactions to occur. However, when we look at a beta sheet, it can be different in terms of the sequence space. For example, we abbreviate the one strand as shown here. Here's your backbone interactions between two different parts of a polypeptide chain, and you can have one chain go here, loop all the way over here, and then come back. So these are amino acid residues. This could be I here, and this can be I plus 38. Okay, so these sheets that are formed between two beta strands don't have to be close in sequence space, but the little stretches of strand here are contiguous amino acids. And so when we look at the hydrogen bonding pattern that can exist between two strands, there are two types, and it's based on the directionality of the strand itself. And that's shown here in the blue arrows. So let's look at the case on the left here. So we're looking at the strand directionality of our polypeptide chain. The arrow is going to point to the C-terminal end. So we go from N-terminus to C-terminus, N-terminus to C-terminus, N-terminus to C-terminus. So this is the direction of each strand. Okay, don't worry about residue number here because it could vary quite a bit. Alpha helices is not the case, but here it could be because there's a lot of other things it could be doing over here before it comes back around. So if you look at the hydrogen bonding, every amide hydrogen is bound to a carbonyl oxygen through a hydrogen bonding interaction, but it's between strands. These are angled hydrogen bonds here, and the strands are going in a parallel fashion, N to C, N to C, N to C. This is called a beta sheet, but it's a parallel beta sheet. Notice this one here, N to C, N to C, N to C. One goes from left to right, one goes from right to left, the next one goes left to right. So these are anti-parallel because they're not going in the same direction. And our hydrogen bonding pattern, because of this anti-parallel arrangement, has a little different hydrogen bonding, or more straight on. So that confers little different structural differences between what we call a parallel beta sheet versus an anti-parallel beta sheet. Okay? We can represent each strand here with this arrow ribbon, and the arrow is pointing in the C-terminal direction. So if you see, and we call these topology diagrams, and you can see here that the arrow is pointing to some kind of a connecting region and allows this other strand to go here. So the arrows are going in opposite directions, so you know that's anti-parallel, and you're forming hydrogen bonding, and it looks like this. Notice the side chains, though, are sticking out above or below the strand. So in the alpha helix, they're sticking out away from the helical axis. Here, they're above and below the strand. Okay, so let's look a little bit more details on some different structures. Here's the parallel beta sheet showing you the detailed hydrogen bonding interaction. Here's a space filling model. If we were to look on the side of this sheet and just look at two strands, you can see how they're arranged, where your alpha carbons are here, here, and here, and you can see their side chains alternate. So once it's down, then it's up, then it's down, then it's up. And the hydrogen bonding interactions are occurring between strands like this, between the amide hydrogen and the carbonyl oxygen. And so you basically have a little sheet here, right? You can imagine your peptide bonds that are formed here are kind of going in these pleated sheets, like if you took a folded piece of paper, and at the vertices you have side chains going up on the top or down on the bottom, and so you can see how they're aligned. Okay, and these sheets in the parallel arrangement have a little twist to them, so the sheet's not completely just up and down perfectly, but there's a little twist to this particular sheet. Okay, so that's the parallel beta sheet. The anti-parallel beta sheet is different in that you don't have this twist anymore, because the hydrogen bonding interactions are such that that twist is not prevalent. But you still have the R groups going up, down, and up, just like you do in the anti-parallel beta sheet. But the orientation is different on strand direction. Okay. Sometimes you have a mixture of sheets that have some anti-parallel characteristics and some parallel characteristics, and we call that a mixed beta sheet. So it may not be all parallel or anti-parallel, but it could be mixed as well. So you do see the mixed sheet with parallel and anti-parallel components. If we can compare, say, a beta sheet, which is two beta strands shown here, to the alpha helix, you can see the distance, say, between your R groups that are going down or to the right, to the left, and to the right, and a scale here. You can kind of see how the 3.6 angstrom difference between the R groups here and here are arranged, so they're very similar almost in terms of their spatial separation, but it's not in terms of residue number, it's much less, it's 2 versus 4. And so again, the R groups are going to be extending above and below the sheet, okay, in an alternate fashion. The hydrogen bonds are perpendicular to the strand direction, and the alpha helix, the hydrogen bonds are parallel to the strand direction, and the side chains stick out away from the helical axis into solution. Okay, so there's side chains that are going to be sticking out over here. You'll have two sides to this sheet, residues on one side, residues on the other side. So those are the structural features that I want you to remember between alpha helices and beta sheets. And these R groups can be such that they can all be hydrophobic. And so you have a very hydrophobic helix. You could have a sheet here that could be very hydrophobic, or one side of the sheet can be all R groups that are hydrophobic, and all R groups on the other side of the sheet be hydrophilic. So you can imagine their interactions would be different. One would be more of a hydrophobic phase, and one would be a hydrophilic phase. So this is important in terms of how these proteins will fold and interact with one another. So know these key features, and I like this diagram that shows you the summary of those features side by side between the alpha helix and beta sheet. Now, proteins will sometimes be all alpha helical, all beta sheet, or a mixture of both. And in addition to that, there's connecting elements that connect these secondary structural elements together. And so this is important when you have to change direction, for example, between making a parallel beta sheet to an anti-parallel beta sheet. And so these turns could be very ordered, or they could be very flexible. It just depends on what the amino acid sequence is for those transitions. And so there are basically two types. We don't have the time to go through the subtle details here of these features, but just know the two types. One would be non-structured loops, so they can be quite long and have a lot of conformational flexibility and rotations about their psi-phi angles. And so there's not really a defined structure. There's not a lot of hydrogen bonding that's occurring there to keep them into a certain conformation. And so they'll have a tendency to adopt multiple conformations, and may adopt a more stable conformation if it's a binding site for a particular ion or metabolite. And you can see how these innervating loop regions are connecting these strands of this beta sheet, and hopefully you recognize this beta sheet as being what? Parallel or anti-parallel. Look at the arrows. Here's the arrow goes here, then back up, then back down. So they alternate direction. It's anti-parallel. And then you have a helix going across the top of that sheet as well. And if you see this two yellow spheres here connected, usually yellow is sulfur, is showing a sulfur-sulfur bond, so that's a disulfide bond, and you should recognize that is from two cysteine residues. So it gives conformational stability as well, because it locks this interaction conformation in place. Now there's smaller transitions as well, and those are called structured beta turns, and they're usually going to be 4 residues in terms of their amino acid composition, and there'll be some hydrogen bonding interactions between residues 1 and 4, and because you're making a drastic change in direction of the strand, so if we're going from here, from the N terminus, making a quick turn and going in this direction to make like a part of a anti-parallel beta sheet with these 2 strands, this turn is about as tight as you can make it. You're not going to get any smaller than this with 4 residues. So there's many turns based on residue composition, and you need something that turns very quickly. You have small side chains, and can you think about what amino acid residues would be good to make drastic turns? What side chains would help bond angles to turn in such a way that you can change direction rapidly without interference from the side chain? I'll give you a hint. These are the same residues that aren't usually found in helical regions. So it's based on their structure. So I want you to think a little bit about that as well. So you can tell by the structure why that would be the case. Okay? So just know the difference. There's usually going to be connecting sequences between these secondary structures. They're either going to be non-structured loops that can adopt a variety of conformations, or defined turns that do have at least 2 hydrogen bonds to help stabilize them to make sharp turns. Hence they're called beta turns. Particularly when you're looking at anti-parallel beta sheets. So when looking at a protein structure, there are a lot of psi-phi angles because there's a psi-phi angle for every residue. And so how do we kind of look at all these angles together to get an overall picture of the protein structure? And so the idea came from Ramachandran, and this is the Ramachandran plot, where you have a plot of your phi-psi angles, as shown here, and it goes from 0 in the middle to plus 180 degrees to minus 180 degrees. So you have the full 3-dimensional space there for each angle of 360 degrees of rotation. And when you're looking at protein structures and start to map these secondary structural elements onto a Ramachandran plot, you'll see that they'll group together and cluster together based on their secondary structure. So for example, if you have an alpha helix, and the alpha helices that we're looking at are right-handed because of the twisting action of how it rotates. Most of them are right-handed alpha helices. They're going to resolve in this region of the Ramachandran plot with a negative phi angle and negative psi angle. 310 helix has a little more flexibility, and so it'll expand out further than an alpha helix would. Above the phi angles for sheets are going to be, or psi angles for sheets are going to be positively valued, whereas the psi angles are still negative. And so you can see your parallel, anti-parallel sheets are in this region of the Ramachandran plot. And you've got some different turns, so type 2 turns kind of reside here because they have those features as well. There's also a left-handed helix that will rotate in the opposite direction, and so it's almost as though you're taking these angles and you're flipping them the other way. So if it were negative, they'd become positive, and that's kind of what happens over there because you're turning the helix in the opposite direction. It's like a screw. You can rotate it clockwise to tighten the screw or counterclockwise to untighten it, but if you reverse the thread on it, it would be the opposite direction. And then you have some other types of turns here that can also exist in some different regions. But notice most of the regions here are devoid of any possible psi-phi angles. And so this is a useful tool to look when you're determining protein structures, because you can plot your psi-phi angles onto a Ramachandran plot and see if you have any psi-phi angles that appear in some unusual region. So if you had something up here, it's like, am I missing something? Am I misassigned something there? Is that part of a anomaly that in my measurement? I need to go check out those residues and see if that's really truly a structural component that's novel and new for this protein, or did I make a mistake? And so sometimes you can get things outside these values, but you want to confirm what those are. And so on the right shows kind of like real data, and you start seeing some things clustering out in some unusual areas, or there's some unique turns that are very well defined in the electron density map of a diffraction pattern that was collected, or is that just some mistake on assignments there? Was it a unique fold or a new fold? So you can get things outside the Ramachandran. Some things in the Ramachandran plot that really can pinpoint some unusual characteristics of a protein. So that's its usefulness as well. Because if you get something outside here, you wonder if you made a mistake or it's just some new folds that are possible. So it depends on the protein that you can get some things outside the normal values here that may be related to its unique structural perspective. So that's the Ramachandran plot. Plot the phi-psi angles. So that's your quick introduction to secondary structural components. We could spend a lot more time with this subject, but as this is a survey course, we want just to present the key details of the two different types of secondary structural elements, alpha helices and beta sheets, and understand the two types of sheets being parallel and anti-parallel. So know their basic characteristics as well as for the helical regions, and that they're connected either through flexible loops or defined beta turns. And as far as the primary structure is concerned, it's just a sequence of amino acid residues for the protein. And so just keep that in mind when you're asked to identify what is the primary structure of a protein. It's just its sequence. We usually just say what's the sequence in the protein, but sometimes they'll say what's the primary structure, and you're like, I know secondary structure, tertiary, what's the primary? It's just the sequence. So work on the problem sets. I'm trying to illustrate some of the key features I want you to know and some problem solving as it relates to protein structure. So work on the problem sets and do the mastering assignments, and this will become clearer as you do those exercises.